first session, I really want to talk about the major advancements that have happened to make deep learning so useful to protein design, and 3 different tools that we'll be using in this workshop. So deep learning has really transformed the landscape of protein design. And I think there's a few different reasons for this, but mostly it's the data that's been being collected for almost decades up to now. So modern deep learning models can be trained on millions of structures and sequences, and they sort of are able to learn these underlying patterns and relationships between amino acid sequences, structures, and even function. And this kind of allows them to understand the grammar of proteins at this fundamental level. Another really nice thing is that unlike traditional methods, deep learning methods can kind of generate new protein sequences that never existed while still maintaining the physical and chemical properties that are necessary for folding and function. And this has been hard up till now. And finally, thanks to breakthroughs like AlphaFold, we're able to get really, really accurate predictions of structure. And so we can kind of evaluate designs by predicting structures before synthesis. So you kind of get an idea of what your protein looks like pretty accurately. So the first major breakthrough that I want to talk about is the transformer. So this is a deep learning architecture. It really excels at processing sequential data. So it was mainly used on sentences before it was kind of applied to protein design. But the reason that they work so well is this self-attention mechanism that transformers use. And this kind of allows the transformer to look at each different amino acid position, and they all get to talk to each other. And so what attention is the sentence or the amino acid sequence is broken up into these tokens, and these are each considered sort of like a node, and they kind of talk to each other. And with the self-attention mechanism, the different amino acids can kind of figure out how much they need to pay attention to the other amino acids. And through this sort of message passaging with each other, they know, okay, this one seems to be closer to me, I should pay more attention to it. And so the matrix of probabilities is changed based on these different interactions. So why do we think attention works so well for protein sequences and structures? So the model can kind of learn which amino acids are likely to be more important. Yeah. So for me, I think attention is so valuable because proteins kind of come together with these long-range interactions. So the amino acids that are close to each other in sequence might not necessarily be important, or there might be further away amino acids that are more important, and attention can kind of capture these long-range interactions. I'm going to jump into AlphaFold and how the transformer is sort of the basic building block of AlphaFold. So like I talked about, self-attention is kind of able to capture these complex relationships between different amino acids. And AlphaFold works this into its pipeline. So AlphaFold works in these 3 main stages to predict the 3D structure of a protein. The first is sort of this input processing and initial analysis section. So it starts off with just the protein's amino acid sequence, and searches through genetic databases to find related protein sequences that are used to create a multiple sequence alignment. And then it also looks for known structures that might be similar, and it uses these as templates. So this gives the model sort of evolutionary context as well as structural hints of what the protein is going to have to look like. So once it has passed through these first 2 stages, it creates the MSA representation and the pair representation, which are both passed into the evoformer. So this is the main transformer. It has 48 layers, and this is sort of the core processing of the model. So it takes the MSA representation and sort of uses it to understand how the protein has evolved. And it also takes the pair representation and uses it to figure out how different parts of the protein might interact with each other. So these keep updating each other through the evoformer, and that gradually builds a better understanding of what the protein structure should look like. And then there's also this recycling process where the model kind of takes its best guess and passes it back in to sort of refine it through passing through the model again. So once we've gone through the evoformer, out of the evoformer come the single representation and the pair representation that have been sort of talking to each other, so they have more information about each other now, and that goes into the structure module. So the structure module basically takes all of these n-dimensional arrays full of information and converts it into actual 3D coordinates. And it uses 8 different specialized blocks to gradually refine the structure. And then finally, it outputs the final 3D structure using, and it also gives you these confidence scores for how accurate each part of the structure is likely to be. So I like to think about AlphaFold as this sort of person that's working on a complex puzzle. It's sort of looking at all these different pieces of evidence and making connections and gradually building up to the final solution. And it takes multiple cycles of processing to get to this sort of finalized confidence structure. I want to talk a little bit about how you can modify the AlphaFold inputs to kind of get the best results for your specific problem. So the first thing that you can kind of mess around with is the number of recycles, and this is, like I said before, the number of times that you're kind of refining the model or the structure through the model. So you can increase the number of recycles. I think in AlphaFold 3 they actually end up using 20 recycles as the default. So this obviously takes more computational time, but if you're going for the most confident, most accurate prediction, then you probably want to crank up the recycles. On the other hand, if you're running a lot of sequences through AlphaFold and you are kind of trying to minimize the computational time, you can kind of reduce the recycles, especially because the higher confidence regions tend to not change much between recycles. It's the lower confidence regions that you can kind of sample more. And so if you're thinking about like conformational modeling or something like that, then that's definitely something to consider. But I usually stick with like 1 to 3 recycles. And then the next one I want to talk about is the MSA depth. So the multiple sequence alignment is sort of crucial for the prediction accuracy, especially for like bigger proteins with like different folds. So a deeper MSA with thousands of sequences is obviously going to be better for AlphaFold to create a more confident prediction. But one thing I have noticed is that if you're working with templates, if you give it too deep of an MSA, it sort of doesn't take into account the template. So if you're trying to get AlphaFold to really listen to the template, then you want to reduce the depth of your MSA. You can also change the way you sample the MSA to sort of get different conformations. So there's the subsampling procedure that you can look more into. If you want to have a really deep MSA, you can put in like every similar sequence that ever existed, but you can also make the MSA shallower by sort of, exactly. So you can like cluster by similarity and only give it like the centers of the clusters. If you want to like get it to predict a specific conformation, you can give it one specific cluster. So yeah, there's ways to play around with that. And then yeah, templates, they're sort of existing structures. So this is sort of like a homology modeling in my eyes. You can provide your own templates, or you can let Colab search like kind of find the templates. And yeah, the key here is really finding the right balance with the MSA so that you're getting the correct amounts of information from both. And then random seeds sort of control the starting points for the predictions. So if you vary these seeds, sometimes you can explore different conformations of the protein structure. So similarly here, high confidence regions aren't going to change much, but it's kind of the lower confidence regions that you can sample more. And you can see in, I think, the AlphaFold 3 paper, for harder problems, they actually sample like close to 1,000 seeds and then take like the best few structures for their actual prediction. So if you're working with something that's a harder problem, for example, antibodies, it's a good idea to run a lot of independent seed predictions and then sort of compile them and find the best one. And then just to briefly go over what AlphaFold is outputting, of course, it's giving you the PDB file with the coordinates of the structure. It also gives you this local confidence metric called PLDDT, which is a per residue confidence that comes from the model. A lot of people will do like an average PLDDT to see how well their model scores. And then PAE is a pairwise residue confidence prediction. So in this case, I'm showing the PAE heat map of a target protein A with a binder B. And you can see a lot of the residues of the target protein with the other residues of the target protein are pretty confident. And similarly for the binder, it's sort of confident in the structure of these two domains by themselves. But when we get to the interface between the residues of A and B, both down here and to the left up there, it's less confident. So that kind of gives you an idea of which parts of the structure or the interfaces. So the PAE matrix isn't symmetrical because it is measuring the PAE by aligning one residue and seeing how much the other residue has an error. So what I usually do to handle this is just average over PAE of residue X with respect to Y and Y with respect to X. Yeah. And then it also gives you, if you're using the multimeric version of AlphaFold, it also gives you this, these two global metrics. So PTM is kind of the accuracy of the overall structure of the complex, and then IPTM is the accuracy of the predictions of the interface between the subunits. And I think there have been recent anecdotal evidence that like sort of optimizing your protein-protein interfaces so that the IPTM is high translates well into experimental success. So some of the things that we will be looking at are PAE, IPTM, and PLDDT. IPTM is sort of telling you how the different domains interacting, how are the different, or how confident is it in the domain interactions. So I'll say they're pretty correlated. If you have lower PAEs, you will have a lower IPTM in general. But if you're looking at very specific regions of the protein, I suggest using PAE over IPTM. So because you can look at pairwise residue confidences at the specific interface that you're looking for and not sort of all the interfaces of the protein, which is what IPTM is kind of telling you. To me, it's like predicting how accurate. It's telling me how confident it is in the accuracy of its prediction. So AlphaFold2 tells you that it's confident in something when it probably has seen something of a similar pattern before, so maybe not necessarily the exact residues or the exact interface, but it's seen that sort of pattern or like interactions of residues before. It is possible that it's missing out on things that it hasn't seen before that are still good, so that's kind of how I would interpret that, is if you have something successful that has a high PAE, AlphaFold maybe just doesn't capture it. For me, I've had a lot of success with listening to AlphaFold, I guess, because like I said, like it has seen successes in the PDB database, so you can kind of use it to filter your designs towards things that work. But again, we might be throwing out things that work that AlphaFold doesn't think are going to work. So yeah, it's kind of just different approaches. So the next one I want to talk about is graph neural networks. So this is sort of an architecture where nodes are connected by edges, and these nodes can either represent residues or atoms, depending on which level you want to be looking at the interactions. And so these nodes exchange information with their immediate neighbors and send messages that contain relevant data. So this might be sounding familiar to you. And I think one thing that isn't really clear to me is what the differences are between like a transform, or like, I think all of these words kind of, you can use them interchangeably. But so we're going to talk about graph neural networks because ProteinMPNN is a graph neural network. And what makes this message passaging so elegant is, I feel, how it mirrors the physical reality of proteins. So just like an amino acid interacts with its neighbors and influence each other through various interactions, the message passaging allows the residues to sort of communicate that with their neighbors. And they can share information about chemical properties, like spatial relationships, and even long-range interactions. So I think that talking about ProteinMPNN as a graph neural network is really intuitive for me, because I can think about how the different residues have these features of coordinates of the NC, alpha, and C. They also include a carbon beta, which is like a virtual atom that they're calculating based on the positions of the backbone. So this is just like maybe a imagine it as like a center of mass of where the residue is, side chain is going to be. The main network has two subnetworks. So first is this sort of encoder. It computes these refined node embeddings. So it takes the structure and basically parses it until it comes up with this like n-dimensional matrix that contains all of this information about the different nodes and how they interact with each other. And then there's a decoder that takes these embeddings and parses it until it can predict a probability distribution at each amino acid. So the major change in ProteinMPNN from the previous model, which John Ingram had developed in 2019, was that they changed this sort of fixed left to right amino acid decoding to a random decoding. And that was really useful because it sort of allows to, it allows inference in situations where you're not just predicting the whole amino acid sequence, but if you're like masking out different regions or if you have some fixed regions, this kind of works better. And then one thing that I really like to remember about ProteinMPNN is that it's trained to predict a probability distribution, and then the loss sort of pushes the probability distribution towards the true distribution. So, okay, here's a question for you guys. What would the true distribution look like in this case for a specific amino acid? So if you have, say, a position where you know that it's supposed to be a glutamine, how would you represent that probability distribution? It would be 100% bar on glutamine and everything else is 0. So it's kind of trying to take the prediction that ProteinMPNN gives it and push it towards that true distribution. And I like to remember that because sometimes ProteinMPNN can, so when it makes the prediction of the amino acid, it's sampling from this distribution. So there's always other options at that particular position. So if you're thinking, okay, MPNN says this particular residue goes here, but I don't really agree, I like to go into the probability distribution and see what other residues it is predicting for that location. And then finally, I'm going to talk about diffusion models. These are models that kind of learn to interpolate between a data distribution and noise. So in this case, your data distribution is dog pictures. You take your data and you sort of add noise to it until the distribution becomes complete noise. And then to train your model, you take the noising trajectories and sort of train it to go from this noisy picture to the original picture. And when you're doing this in inference mode, you can give it a random noise distribution, and it'll sort of create something that should belong in the original data distribution. So it won't necessarily match something in the original distribution, but it will sort of have the properties of the original distribution. And so, of course, we're going to talk about RFDiffusion. And you can see that sort of gradual process of going from noise to a protein structure here. And the main thing about RFDiffusion that I want to point out is that it's built on Rosetta fold. So RFDiffusion is kind of using Rosetta fold's structural knowledge that it's already been trained on to make informed decisions at each step of this denoising process. So this is what a step looks like. It goes through Rosetta fold, and then so I like to imagine this as if you have a noise distribution, it's kind of like you have this giant chunk of clay. And Rosetta fold here is an expert sculptor, and it's telling you how to guide your hands. And so Rosetta fold diffusion is sort of just pushing the clay into something that the expert sculptor says, okay, make this change, make that change. So that's how I like to imagine it. So you can see here the gradual sort of denoising and how it becomes more and more protein-like at every step. So Rosetta fold is a model that's kind of similar to AlphaFold. It does have 3 tracks, so it has the MSA, pair, and template track. But I would think of it as like similar to AlphaFold for this case. Cool, and so since the input to diffusion is just a noise distribution, we can kind of modify this noise distribution to make it do more useful tasks to us. So if you give it just Gaussian noise, it's going to be able to generate an unconditional, so this is called unconditional generation. It'll create like a monomer of a protein backbone. If you want it to generate, say, a symmetric protein, you can give it symmetric point clouds and ask it to denoise these. And similarly for binding targets, functional motifs, and symmetric motifs, you can give it these fixed positions as well as the noise and just ask it to sort of denoise around the fixed positions. And so that's how we have been able to extend it to all of these different applications. Cool. And then the last thing I want to talk about here is the potentials. So the potentials really help to guide the diffusion process, and I like to think of it as they're kind of pushing it in a specific direction. So you can see here the sample function without a potential and with a potential. The only difference is this sort of guiding scale, and so that's kind of pushing the denoising in a specific direction. And so there are multiple types of potentials that are useful. We'll play around with these a little bit in the activity. But so these are some of the inbuilt ones, and then you can also build in your own potentials if you're feeling like these don't cover what you need to do. Okay, that's the end of this short lecture.